Greetings. I am Stephen Gager, a biology instructor at Ball State University, and this is a talk that I presented to the Science Circle, a science education group that meets in Second Life. The talk Moths, Black and White Proof of Evolution, is going to cover two topics. One is evolution in a nutshell, in which I will be using slides modified from Pearson Biology in the textbook Campbell Biology, ninth edition, which my university has adopted. And I will then also present information about an interesting, I think, parable in which a largely settled science question became a centerpiece of controversy, partly because there are uh, processes in science that can be misrepresented in a way to seem like there are issues in which there in fact are none about the basic science. And I think there's an interesting story here about a scientist who really dedicated his life and research to addressing something that really did not, in a sense, need to be addressed, but I think does illustrate some important aspects of how science is done in modern days. So to start off my evolution in a nutshell, I'm going to present Darwinian evolution, which, and again, Campbell ninth edition is chapter 22. So Charles Darwin published in 1859, again, the full title, On the Origin of Species by Means of Natural Selection or the Preservation of Favored Races in the Struggle for Life. And this was a treatise based upon decades of work and observation about how species come about, and something that was not scientifically explained at the time. That Darwin had the unique insight to think about how adaptations to the environment was essentially the causative reason for why species would vary. Now, he did not actually use the term evolution. And his theory of evolution really is a combination of two separate, in a sense, theories. One is descent with modification, in which it says essentially every single organism that lives today is the product of something that came before it. And again, this would be in opposition to the prevailing view at the time that species were all just created, that they were spontaneously made by divine intervention. The other component of evolution is natural selection, which is also sometimes referred to as survival of the fittest, but in essence, that within a species there are individuals who uh, not only survive better, but produce more progeny with their favored genetics, uh, and that those tend to become the population or find ways to branch off and become new populations. So again, this is in opposition to the idea at the time that all species were specially created and adapted and perfect for the environment in which they live. And because the ability to have variation, and that's acted upon by forces where some are favored versus the other, that's not about being perfectly designed and created. And I will give a little bit more description about how these processes work, but I'm really going to give a very short form version of it that is sufficient to set us up to understand a little bit about how moths were studied to understand and use, in essence, they were coined as proof of Darwinian evolution. So this is not a complete story of evolution, but enough to get us understanding where we need to go. Now, I would be remiss if I did not mention that one of the advantages, or again, those happy circumstances that led to great discoveries that Darwin had, was an opportunity to take and be a scientific advisor and captain's conversation mate on the HMS Beagle. And this was something set up for him by a colleague and mentor partly because Darwin essentially failed out of medical school. He had essentially no constitution for it. And so the fact that this was a 5-year survey and he had lots of time to collect specimens, ruminate about his observations, is one of those reminders that great discoveries are usually a combination of a great intellect as well as opportunity to exercise said intellect. But it's also worthwhile to point out that if one reads Origin of Species, that a lot of the compelling arguments and data were based upon decades of hard work by Darwin looking at domesticated animals, his own breeding experiments with pigeons, and interacting in various social circles with the intelligentsia of England at the time. Of course, the most famous stop that is related to Darwin's journeys is the Galapagos Islands, which are small islands, again, on the equator to the west of South America. I mean, I think it's worth noting, if one looks at historical records as well as Darwin's journals, that there were many places along his voyage that provided seeds and observations that helped him develop his theory of evolution. But the Galapagos Islands are again, the one that's most famous and familiar with many people. And so I'm going to take one example of the animals he looked at from there. And that would be the Galapagos finches. And these were birds that Darwin observed on the island. And from the examples we have here, they each had distinct behaviors and morphologies. And again, the key thing to point out here is that the cactus eater, the insect eater, and seed eater each had very distinctive beak shapes, as well as probably the jaw strength that was consistent with the animal's observed food source. Now, it turns out Darwin did not actually identify these each as being finches and the other species originally. He was actually not an ornithologist, but he collected the samples, and it wasn't until he got back to England and someone identified and said, oh, these are actually all finches, it struck Darwin as inconsistent with the idea of special creation. So, for example, why would various other mainland habitats have a wide variety of birds, not just finches? And why would there, if there's only finches on this one island, why would they have such a wide variety of morphologies and, again, essentially distinct species? Because you'll notice that not only do they have different beak shapes, they look very different in terms of coloration. And so it struck Darwin that really the only possible explanation for why these are all finches is that they were all derived from some original finches that somehow, because they are flighted animals, were able to be displaced from the mainland, settled here, and then must have evolved over time into the wide variety of distinct and different species as well. And so from this example, among many others, Darwin eventually proposed this idea that species arrive from, again, previously existing species, and then there are descendants that have modifications, and then these go on to, again, ultimately become new species. And this is visualized in Darwin's journals and the Origin of Species as this tree of life, this branch point where a progenitor animal, again circled number 1, goes along, and then different conditions over time create, and in different or perhaps similar geographic regions, create these branch points that lead ultimately to new species. But of course, the drive, the reasons, the cause for why these new species do develop is something that is the second half, the natural selection part of Darwinian evolution. And so the special term for the creation of new and distinct species from progenitors, that's known as macroevolution. Again, evolution on the scale of the big. And what I think I do want to point out here is that the rest of my talk and talking about the moths, those all are examples of so-called microevolution. And that is not where you have distinct and new species being created, but selective forces that are similar to what happens in macroevolution that lead to changes and shifts within a population. And so the forces of selection that Darwin described had to do with one, artificial selection. And that'd be examples of uh, breeders, people that either with plants or animals have species, and then within those populations of species they're dealing with, they say, "Oh, I'm going to keep this individual and get rid of these other ones." So, again, horses that are faster, or for horse racing, or horses that are stronger for being beasts of burden. Flowers that have distinctive colors, or even examples that he favored and did himself was pigeons. Again, pigeons that had different colorations or different traits. So uh, those would be selective forces. Again, somebody intervening and saying, "Oh, these individuals are better than those," but that's called artificial selection because it's something that humans are directing. Now, within wild populations, the argument he made, which was based upon and had some influence from Thomas Malthus, who wrote an essay on the principle of population. And in that, Malthus held that populations and individuals can reproduce geometrically, and that if they're allowed to reproduce unchecked, they will eventually use up all of their natural resources, be it the ability to conduct agriculture, or just the amount of available food, or even the amount of geographical space one has. And so that means if people are running out of space and food, then within a species you'll have to compete. And so it's those individuals that can compete better and produce more offspring over generations that influence Darwin's thinking. That again, within a species, you're always competing for resources. Again, even before you get to the point that you might be starving. And so these influences mean that, again, if you have some individuals that have variations, then those will be favored over subsequent generations. And so that was the idea that within natural selections, though those individuals that survived more often or produced more surviving offspring would then, as a proportion of that species, become more prevalent. And if these variations accumulated enough to the point, then you could actually find examples where that would lead to, again, distinct, different looking individuals and be distinct from the original population and species, and that would eventually lead to the generation of new species. So it's this combination of individuals that pass on variable traits to offspring, and then they're all competing for passing those genes on from generation to generation. And so this schematic is a nice summary of Darwinian evolution. And again, the two selective forces and the variation that we saw at the top, these are the forces of natural selection that drive traits to be favorable within an environment. And that's the key thing, is that uh, the one of the prevailing thoughts was that species seem to be perfect for their environment, and the Darwinian evolution with natural selection favoring traits that are favorable to the environment would lead to the same thing as divine creation that makes them perfect for their environment. So again, a very separate explanation for this phenomenon that had always kind of puzzled people. Again, I think it's important to note that Darwin said there are lots of reasons why I believe we should not consider organisms perfectly suited. Again, the idea of vestigial organs that we don't use anymore. That seems contrary to being perfect if you have something that you don't actually ever use. But it makes sense if it was something that an ancestral organism did use at one point, and then because environmental or other conditions had changed, it was no longer useful and thus tended to degrade in structure and form. And the other key part and context that we should think about with Darwinian evolution is that for species to come about via this process, it would take a lot of time. And a lot of people believed the Earth was very young. And so, again, Darwin's willingness and understanding that the Earth was, again, he thought millions of years old, which we now know it's actually billions, was something that had to be present in order for his view of descent with modification to lead to new species. And another thing that's very important to note is that Darwin actually knew nothing about molecular biology because the field had not been invented at the time. That he was working his theory off of very observational data, but did not know about DNA, did not know about the chromosomal inheritance, all things that we take for granted today as a part of how we understand the specific chemical and life-based uh, inheritance of traits that come down to the molecules involved. And so, speaking about people who were very important in our modern synthesis of how we understand evolution is Gregor Mendel. And he was an Austrian monk who was a dabbler in various scientific pursuits. One of them was to look at the heredity, the inheritance patterns of pea plants. And this was something he did in his spare time when he was not otherwise being a monk. And of course, now he's known as the father of all genetics. And I do want to point out that we have found very many other types of inheritance patterns that we actually just call non-Mendelian. But to illustrate what we're going to do with the moths later in the talk, we actually only need to understand specific Mendelian inheritance patterns. And so what I'm going to provide here is a summary of Mendel's work, looking at one specific example of the 7 different traits and characteristics of pea plants they looked at. We're going to look at the color of the flower. And so some of the terminology that's important to talk about is at the very top here we see that there are pea plants that have purple flowers and those that have white flowers. And the term true breeding is a term meaning that a stock of pea plants with purple flowers, if you breed them amongst themselves, every single individual over time is purple all the time. And that would also be true for the white, that if you have a batch of white flowering pea plants, they are always white. And what Mendel did, again, in a controlled fashion where he could do things like uh, bag the flowers so they didn't get accidentally fertilized by a plant he wasn't aware of, or he would also trim off the stamen so that they could not accidentally pollinate something else or self-pollinate, that if you bred those 2 different pure breeding plants, then every single individual in that F1 generation, the hybrids, they were always purple. This is in contrast to what many people might expect, is from a blending perspective that they would be some sort of lavender color. Again, the idea about evolution, even the one that Darwin was working off of, was that the characteristics of 2 individuals that led to progeny was a blending of their characteristics, which actually is a nice description of more complicated genetic patterns, but it's actually a combination of these types of discrete patterns that Mendel ultimately characterized. And again, what I mean by discrete, in terms of discrete elements that cause the phenotype, again, the phenotype being the coloration, is that when he took those F1 hybrids and bred them amongst themselves, either self-pollinating or cross-pollinating, that this white coloration would come back. And the ratio of this white coloration was 1 out of every 4 individuals. So there's a 3-to-1 ratio of the purple to the white. And so again, if you had a blending type of inheritance pattern, then it makes very little sense that flowers that are completely purple would somehow occasionally spit off something that is a completely different color. And so what this meant to Mendel was that there are discrete elements that are the instructions for making the color, and that their interaction must have some sort of rules to it that lead to ultimately the phenotype of what you see. Now, Mendel's really important contribution to this is developing a nomenclature that helped him describe and then ultimately allowed people to work together to describe genetic phenomena, where the idea here of one trait uh, being the dominant trait in that F1 generation, the purple being the color you see when you mix a true breeding purple with a true breeding white, meant that it was dominant, and he gave that a capital letter, so a capital P for purple flower, and that the white flowers, which were recessive, which he termed recessive, was a lowercase p. And that what must be occurring is that any given flower has two copies of the genes, but when it passes it on to the next generation, it's only giving half of it, which makes sense that if individuals of species were always passing on and mixing their genes with another individual, if you didn't reduce it down to half in the first place, then you keep getting more and more genetic material in the progeny, which makes very little sense. And so there's this reductive process where if you start with two copies of something, each gamete that you're passing on, like the pollen or the egg, is half of that. And that these are discrete elements that are, of course, always passed on. You're always passing on a so-called gene that encodes the color of the flower. And so what made sense then is that if that F1 generation was a hybrid of being a purple allele, as indicated by the capital P, and a white allele, which is indicated by the lowercase p, that even if it were self-pollinating, that half of the pollen would have the lowercase p, the other half would have the uppercase p, and that would also be true for all the eggs, that would be half and half of all those. And if you start mixing those categories of half and half, then 1/4 of the time you'd expect those white flowers to come back. And again, the idea here that these are dominant, of course, means that the heterozygotes, again, the ones that are a capital P and lowercase p, they have the same color as the capital P individuals. And so Mendel, having a grasp of probability, what his ultimate theory rested upon was that in looking at these 3-to-1 ratios, was inferring from those probabilities that what must have made them were the flipping of coins, where there's a half and half probability of, again, in terms of the union of a male gamete with a female gamete, that ultimately led to these combinations of alleles. And again, one thing to point out here is that Mendel's theory correctly explains the inheritance pattern of some very simple genes and explains them within one individual mating, the 50-50 chance of what is happening when individuals are making gametes. But again, a very powerful new way of understanding how evolution could occur is on these discrete elements, where again, I like having this picture with the flipping pennies, where one can now start applying the frequency and probabilities and the distributions of these, which is what we'll be talking about in this very next section. And so this last section of evolution in a nutshell is taking those principles of understanding discrete genetic elements and alleles and applying those to population as a whole. So something that gets very, very mathy, although we only need concern ourselves with the basics of it to understand how this was used for the moths. So in thinking about populations and genes, let's just start out with the idea that there is flower color in a plant that the alleles are either red or white. And just from a starting point to talk about this, we will imagine that the frequency of the red allele in a population of adult flowers is 0.8, and the only other possible allele for this particular characteristic is the white, and that would be 20%. So again, that sums up to a frequency of 100%. And that if you just imagine again, zoom your lens way back looking at, say, a field of these flowers, that you're no longer concerned with the probabilities of individual matings being half and half for each one. You just step back and imagine that all the pollen in the air, which again, this textbook likes to use the human gamete variation, the sperm, and that all the eggs that are the ovules or eggs that are inside each individual flower, that those represent just a mix of these frequencies. Again, you're no longer worried about the 50-50 chance in passing it on, but that the frequency of eggs are going to be 80-20%, and the frequency of the sperm will be 80-20%, and that those are the chances of these two encountering each other. And I want to point out that from the population genetics point of view, that one can analyze populations really and look at them mathematically, not in terms of what you're seeing from them or what their characteristics are, is that they really represent populations of genes. Again, what are these discrete factors that define the population? But you can study them mathematically from the point of being genes. And so when a population of individuals is now newly created at next generation, as represented by the flowers that are inside the squares, that the frequency that one will see of the combination of genes that are 80% red from one end and then 80% red from the other type of gametes, that it creates these very specific proportions of the individuals in that next generation. So you'll see here that if you have zero point 0.8 times zero point 0.8 being the red sperm with the red eggs, then 64% or zero point 0.8 times zero point 0.8 is 0.64 or 64% is that so-called homozygous red population. And so what you'll see represented here is that when you have a white sperm allele mating with a white egg allele, which is each of the case 20% of the time, well, 20% of 20% is 4%. And so that combination of those two is going to be the 4% white flowers in the population. And so the remainder of the flowers will be heterozygotes, that is, a white being combined with a red. And so 20% of 80% is 16%, but you have two different combinations. You can have that white allele coming from the sperm, and that would be one set of 16%, or you can have the white coming from the eggs, and that would be another 16%. So in essence, you have a total of 32% being heterozygous. Now the inheritance pattern shown here is non-Mendelian, and it's very useful in this case because the idea here that a red allele with a white allele leads to a pink flower allows us to identify those recessive alleles in individuals that also have a dominant allele, which one could not do with your standard Mendelian inheritance. So again, one thing to point out here is that this is an inheritance pattern known as incomplete dominance, in which the heterozygotes have a phenotype that is intermediate between the homozygous dominant and the homozygous recessive. Okay, so now that we have frequencies and counts and alleles, we can use this to understand changes in those over time. And so let's take the scenario where what we have here is a field of flowers that has a combination of red, white, and pink. And we can actually directly count and figure out the allele frequency of the CR red, this red encoding allele, as well as the white. That's because in this case, we're using one where you have, again, the heterozygotes. And so you can directly count these allele frequencies. And again, for whatever reason, the numbers start out as 70% of them are red and 30% of them are white. Now, again, one convention we have here for alleles is that when we refer to the frequency of the dominant allele, we call that P. And when one refers to the frequency of the recessive allele, we call that Q. One thing that's important to keep in mind is that this only really applies to large populations. If you have a population that's too small, then things can change, and those are just due to so-called random stochastic forces. And so now let's say we come back to that same field the next year, and we do the same thing. We count up the number of red, white, pink phenotypes. We use that to count up the genotypes, calculate P and Q, and we discover in this scenario that they're the same as they were the previous year. And let's say you come back the year after that, count up the flowers and the colors and the frequencies of the different genotypes, then, and you discover that again, the frequency of red alleles is 70%, the frequency of white alleles is 30%, and so that suggests that one allele and those associated phenotypes, there's really no difference in terms of who is going to live or die in terms of those subsequent generations. It really suggests in the end that there is no selection. There is no selective pressure on the alleles. There's no benefit. There's no detriment to having them in this scenario with these flowers in this particular setting. So let's imagine that we have, again, the same flowers, but maybe a different location or a different set of years than we saw in the previous scenario. But let's start them out with the same frequencies of 70, 30%. And again, in this case, we're analyzing the flowers in 5 generations later. So maybe, again, very long-term study. You come back and discover that the frequencies have changed. And that is now P is measured as zero point 0.5, Q is zero point 0.5, so there are fewer red alleles and there are more white alleles. So this change in allele frequency suggests that maybe the red color is bad, or perhaps the white color is good. And again, this is just quantitative. One can just see these changes and say there must be some sort of selective pressure. And then usually it takes some time and investigation to figure out maybe what the difference actually is. For example, maybe red is a more obvious color to a flower-eating animal, or perhaps white includes some sort of color advantage in terms of being able to get bees to pollinate it more often. And so these types of things take more investigation, but in terms of the quantitation, it gives us some understanding or suggestion that something is changing and there's a selective pressure acting upon that population at that time. And so let's imagine another scenario where you have this same starting population of plants. And then within 5 generations, you see that everything is 100 and completely one allele, in this case, red. And so this suggests, again, there must be some sort of selective pressure where red is good, or perhaps white and pink are really, really bad. But one thing that this does illustrate is that notice the overall change in allele frequencies was much faster and stronger than we saw in the previous scenario. So how fast the alleles change can be an indication of how strong the selection is. So this is, and I'm going to summarize here, something known as the Hardy-Weinberg principle, that under certain assumptions for populations, including being large and including that it is, in essence, a closed population, there's not immigration or emigration, and some other assumptions, that we can use this to understand selective processes. And this is known at the bottom, what you see here is the Hardy-Weinberg equation, which is p squared plus 2pq plus q squared equals 1. And what we also described earlier is that if you have a population where the only gene you're analyzing that has only two different alleles, and dominant and recessive, then of course all those alleles must also sum up to 1. So p plus q equals 1. And so this simple equation actually gives us the power to infer things about populations by doing this type of analysis. And so just to reiterate the methodology here, if you want to analyze a population for selective pressures and you know a gene that you can understand its inheritance pattern, that's simple Mendelian, if you have a population, you can just count up the number. And so in this case, we've counted up, we have 64% red, 32% pink, and 4% white, that once you have the numbers of the phenotypes you're seeing, again, and these are the genes being present in 2 copies in each individual, then we can also find and derive the plain allele frequencies, derive P and Q. In this scenario, for example, if we know that the frequency of the homozygous dominance is 64%, in other words 0.64, and that equals p squared, then the square root of p squared is p, the square root of 0.64 is 0.8. And that's also true for the white flowers, that if q squared equals 4%, then the square root of q squared is q, and the square root of 4% is 0.2. And the convenient aspect of that last part is that if we had an inheritance pattern that was your standard Mendelian, again, if we had red flowers for the heterozygotes as well, then one can always calculate Q from the Q squared population and then go back and just do some subtraction from 1 to get P. Now that we have an introduction to the science background, let's talk about the specific organisms that were being investigated. This is the British peppered moth, Biston betullaria, and what you see here is the Typica morph. In other words, it's the typical version or the true version of the moth. And you can see from its characteristics that it is very hard to see on the lichen-covered uh, bark. And in fact, this picture here side by side highlights the moth so that you can see it more easily and directly. The first recorded observation of a distinct variant of the peppered moth was made in 1848, which again is a year of significance in that it is in the midst of the Industrial Revolution in England at that time. It was called the Carbonaria morph because it was very black. And I'll have to fast forward in time here a little bit, but what was ultimately characterized with these variants is that the black peppered moth was inherited as a simple single gene using Mendelian inheritance where the black was dominant over the typica. So if you had a heterozygous black and typical set of alleles, then the moth would be black, like the Carbonaria. Or in other words, very similar to the purple versus white coloration that one saw with Mendel's peas discussed earlier in the presentation. And so this phenomenon, again, the combination of the emergence of a darkly and more well camouflaged variant of an organism, is known as industrial melanism. And it's something that's seen in a variety of organisms where, again, like you can kind of imagine, as the environment has changed, then those individuals with variations in phenotype where it's favorable for them to survive, then they would increase as a percentage of the population. And so what's important to recognize here at this point is that the moths undergoing a challenge in their environment and having genetic variation that seems well adapted to that new change is an excellent area to study using population genetics, studying Hardy-Weinberg to demonstrate an evolutionary process that is occurring at that time. This was recognized by the early 1900s as potentially an excellent case study in order to demonstrate in a quantitative sense evolution in action. So the study of moth populations in England and doing experiments in the field fell to Bernard Kettlewell, who was a doctor at Oxford. And I picked these two pictures in juxtaposition to show one, the professorial looking portrait on the right, but that as you see on the left, he was a man who very much enjoyed getting out in the field and enjoyed getting his hands dirty and directly experimenting with animals in the field, and was, again, prior to his training, a very accomplished lepidopterist. Kettlewell was born in 1907 and went through both medical as well as zoology training, including time in South Africa where he lived with his family. He was appointed in kind of a dual position at Oxford and then became a senior research officer at the Department of Genetics in 1954 at the University of Oxford. And it was this position with the supervisor E.B. Ford where he then conducted the butterfly experiments. Now, why did his work taken towards butterflies is that, and this is not listed here, is that from a young age he was very interested in butterflies and actually was a published lepidopterist by the time he was in college. And so this is a select bibliography of the key papers from the peppered moth work, which included both field studies as well as in an enclosed space studies of direct observations, and as well as his capture-release experiments. And you'll notice the two bolded phrases, evolution in action and Darwin's missing evidence, and these were kind of the key phrases that are associated with his work helping to establish evolution in the way that it had as a quantified and true theory. And I think the one thing that's very important to point out here at this point is that these terms, evolution in action, Darwin's missing evidence, these are bold claims. And so the amount of scrutiny and its importance at the time, as well as something we can look back to as being a very textbook example of evolution, is a part of later in the talk when we think about how people can view this in kind of hindsight. And so this map from Heredity Volume 12, which was published in 1958, is a survey of the percentage of different moth populations that were the Carbonaria, the Typica morphs, as well as an intermediate one, not with different genetics, known as Insularia. And so the higher percentage black you see in those circles, then that is the higher proportion of the Carbonaria, or black morph, in that particular surveyed population. And so if one overlays this map along with the amount of industrial pollution and industrial centers in England, there was a high degree of correlation. So this, again, is kind of an epidemiological perspective on you have more industry, you see more industrial melanism as a percentage of that population. And so that correlative evidence, though, begged the question, really, what was the selective pressure that led to the higher population percentage for, again, the more well-camouflaged morph? And so Kettlewell set out to do several experiments, one of which was to place moths that he bred or captured and place them on trees in an enclosed place, and then observe within a short period of time what happened with them. Because the main idea here was that predation by birds while moths were resting on trees would be the main force of selective pressure. So again, a bird would not be able to, on this particular tree bark, see the Typica morph as well. So at a higher percentage, it would feed upon the Carbonaria. So observing moth predation in a small enclosed place is an important foundational experiment for this, but it's not something that would be generally accepted by the scientific community. And that's because in experimental science, there is this kind of like a Heisenberg uncertainty principle for experiments, where the more you create certainty in controlling your experimental conditions, making it more reduced, limited, but something that you can quantify, that almost certainly makes it so that you are more uncertain as to how that really reflects the actual natural situation. And so performing an experiment like taking large numbers of moths and placing them out in the woods would be, again, another very important step in showing the selective process going on. And so that's what Kettlewell did, was with both pristine woods as well as more sooted and polluted woods, he raised large numbers of moths and essentially placed them on tree trunks so that he had a tally and knew where they were and of which type. And then the next day would count those, as well as put out lamps at night to try and recapture the moths that he had placed out, all of which he had to specially mark so he would know which were his moths compared to the moths that were naturally present. And so this was performed several times in several woods. So this slide with all the numbers on it is an example data set from Kettlewell's papers. And what one would expect, just like we had with the red flowers being 70% red alleles and staying that same percentage over several generations, that lack of change was an indication there was a lack of selection. And so what we saw here in the dead end recaptures, which again is a woods that is considered pristine, and so the Carbonaria morph would be at a disadvantage, would be more obvious to bird predation, is that if you look here on the left set of columns, that's the observed, what number of moths did he actually recapture, and that was compared to the expected value. So on the right, that was essentially the beginning percentage of moths that he released in the first place. And so you'll notice that the number 32 is lower than the number 47, again highlighted by the orange arrows, and so that meant less black ones were recaptured, and that correspondingly the Typica morph was recaptured, 67 were recaptured as compared to the expected number of calculated number of 54. So again, a clear indication that the Typica have a survival advantage in a pristine woods. And the converse experiment at Birmingham, which was a polluted forest, that here, looking again at the left-hand side, the observed number of recaptured carbonaria, or black moths, those having a suspected camouflage advantage, were recaptured at a higher rate than what was expected. And the Typica was recovered, only 18 compared to the expected number of 36 was recaptured. So the fact that the Carbonaria was captured at a higher rate than expected and the Typica captured at a lower rate than expected was again indication that there was a selective advantage here by the Carbonaria. And something that should be noted in that I'm not showing here in the data tables is that Kettlewell did observe a couple examples in the wild of birds rising up, uh, grabbing a moth, and then eating it. So that type of direct observation had not been made before in a well-controlled natural environment. So there you have it. That was essentially considered and promoted a textbook example of evolution where selective forces were known, one could see a change in allele frequencies, and it actually became, for many textbooks, the textbook example of evolution. And so other examples of industrial melanism had been observed, and this was something that a professor, Mike Majerus, at University of Cambridge, uh, put into a book called Melanism. And again, the whole book was dedicated to the idea of changing populations in response to changing environmental conditions due to pollution. And I think one day it would be interesting to do more of a talk about Dr. Majerus, to talk about his background and his research. But for sake of this particular conversation, we're going to focus on what he said about Kettlewell in this book. And to largely summarize some of the main points, that while talking about melanism and those examples, that in looking at Kettlewell's experiments, there of course were some aspects of experimental design that he wanted to point out. And in terms of the interpretation, is bird vision the same as human vision? Uh, is bird behavior changed when one has such large numbers of moths being released at one time? Was the release of the day, where again typically moths will be either in flight at night or figure out where they're going to settle during the morning hours. Could that be different? Were the lab-bred moths different than wild moths? And again, did the placement on the trunks and the additional markings potentially make any sort of difference for the experiment as well? And again, I think this is one thing I want to come back to, that in the normal discourse of science, one does revisit the experimental design of older experiments, sometimes with new technology, sometimes with just a different insight of new knowledge. And this was not meant to be a critique of the conclusion or to say that Kettlewell was wrong or that Kettlewell committed any sort of fraud, that this is a normal aspect of the scientific process for any given experiment. But it does highlight the idea that when, again, we think about specific quantitation of experiments or some of the experimental confounding factors, always good to look at these types of experimental design issues. Nonetheless, that set of criticisms from Majerus ultimately led to the publication in 2002 of a book by Judith Hooper known as Of Moths and Men: Intrigue, Tragedy, and the Peppered Moth. And what the majority of this book does is, from having heard about these criticisms of what was the seminal proof of Darwinian evolution, she wanted to investigate, really, was there something more to that? And a lot of what the book is really dedicated to is more of a biographical set of information about the peppered moths, about Kettlewell, about Ford, about the scientific process, and again, what occurred. And a lot of it is really trying to call into question the character and motivation of these main players and the pressure of, again, publishing in science and associated especially with this particular experiment. And she made a few cases about what sort of maybe fraudulent types of activities there were, but ultimately there was not any sort of real scientific or rigorous investigation as to whether there was fraud, and it really was more making the case that these people had the pressure to do something fraudulent. Nonetheless, whether this was her motivation or not, this became a rallying point and cry and very prominently cited work that for people who did not want to believe in evolution wanted to criticize the seminal example, and that if that was fraudulent, if scientists were making stuff up for their own gain, then that must mean all of evolution is wrong, which of course is a bit of a fallacy in thinking as well. But just to show some examples, so on the website, story titled Creation, Darwinian Evolutionary Frauds, Part 11, with the associated link, said Peppered Moth to a Flaming Fraud. Then another listing 10 scientific frauds that rocked the world at Neatorama. Again, number 4 is actually number 4 on the list, the most unnatural of selections. So Majerus decided to do an experiment that addressed the criticisms he had had of the original Kettlewell experiment. Now, whether this was something that was prompted by the quote-unquote uproar of Moths to Men, or whether it was something he had planned on doing anyway, he did perform the experiment under different conditions. He presented this to the European Society for Evolutionary Biology in 2007. So the upcoming slides are actually derived from his talk, which were available online. And to summarize, the improvements in the protocol were to do it in the wild at low frequency, which also meant during a very long period of time, releasing the moths into natural resting positions, as well as release the moths at dusk so that they would then go to a natural resting spot of their own instead of being placed there, as well as using male moths that were caught or trapped as compared to ones that had been entirely bred in the lab. Additional improvements were using the moths that were originally indigenous to that area, as well as releasing moths at the frequencies that they occurred in the previous year close to the experimental site. And then he actually ran the experiment during the months that the moth is naturally on the wing to account for perhaps different behaviors that were seasonal. And then finally, predation was scored by direct observation or absence after 4 hours. So not a matter of capture release, but direct observation. And he, in fact, of the missing moths, he actually observed about 35% of the predations. And in fact, my understanding of it is that he was doing this outside his house, outside his window. So it's relatively easy to be able to keep track of this over a long period of time. Now, the one thing that he could not do, that Kettlewell did, was the converse experiment of looking at what happened in a polluted environment, because by this time, pollution had been largely cleared up, and there were only the lichen-colored trees to work with. Now, one of the other criticisms of Kettlewell's experiment was that by placing moths on trunks, that was not a natural environment and thus not a natural form of predation by the birds. And so Majerus had done a survey of where he found moths in their natural resting places, and about a third to a fourth were in fact on trunks. So the Kettlewell experiment was not an unreasonable or unnatural predation environment for the moths. And so these are the big results. That if you look at the graph, the blue dotted line represents the percentage surviving of the typicomorph, and the red line shows the percent surviving of the carbonariomorph. So again, a reminder that this is in a pristine environment where the typicomorph would have a camouflage advantage. And so consistently from year to year, there was a difference in the predation rates of the two different morphs, favoring the one with the higher camouflage. Now, one thing you might notice in the publication list in the title is that Majerus's name is in the title, not as one of the authors, because he actually had died in between his collection of data, the presentation of this to his colleagues, and before he could publish this. So this was, again, in many ways published in Evolutionary Biology by friends and colleagues who felt it was very important that his last experiment be communicated to the world. And I suspect, in part, because they also felt that it had this importance and relevance to be something that addressed what were, in many ways, very valid scientific criticisms that made one wonder about the exactness of the numbers from the original experiments, but also, very importantly, to try and silence those people who would like to use the claims of fraud and the aspects of skeptical science against the important contributions in important areas, especially in evolution. So this is the last slide, which is actually from the Majerus talk, and I would like to summarize by saying that I hope you learned a little bit about evolution and genetics and genetic analyses of populations, especially as they change, that it is a quantitative field, one that is amenable to study both in natural as well as in more laboratory-like conditions that'll do allow us to infer evolutionary processes, and that these are something that allow us to say that evolution is something that is occurring, and we have the ability to study it even over short periods of time. The other process of science thing I would like to point out is that scientists themselves are always healthfully skeptical of the work that they themselves do, as well as people before them have done as well. And this is a part of the scientific process and a part of the scientific generation of knowledge. And that this is very distinct from people who will make arguments that something was not correct to mean that everything must have been false or perhaps was fraudulent. So I thank you again for your time and your attention, and that's the end.